It's my pleasure to welcome you to this Ask the Session author. Today we have with us Professor Steinberg, working at McMaster University. He's a professor of medicine, director of the Center of Metabolism, Obesity, and Diabetes. He has many publications, very interesting publications. Please go on NCBI and look at what he published. And now recently, it's a really, really great paper that he published called ECLI Inhibition Promotes Tumor Immunity and Suppresses Liver Cancer, which was published in Nature. So, Professor Steinberg, really congratulations for your very nice paper to you and your co-authors. And I will allow me to ask you a few questions about it. The first one is, as you know, in hepatocellular carcinoma, we have immuno-oncology, we have multi-kinase inhibitors, but we are really lacking new mechanisms of action. And so, how did you arrive, or what was the rationale to target this enzyme, ATP citrate lyase, to treat hepatocellular carcinoma?
Okay. Well, thanks so much for having me. And very nice to meet you and chat. We've been working on fat metabolism and liver for many years. And we know that liver cancer is rapidly rising worldwide due to the incidence of fatty liver disease or MASH, metabolic dysfunction associated steatohepatitis. And so in this disease, chronic fat accumulation and inflammation change how the liver uses energy, making them more likely to turn cancerous. So this is very different from viral-driven uh, HCC. And so these tumors that develop in this context are also very different metabolically and immunologically. And they resist therapies such as immunotherapy. So we needed new strategies really to reprogram both the metabolism and immunity inside the tumor to make the therapies work better. And this was really the principle for studying ATP citrate lyase in this context. Uh, so ATP citrate lyase is an enzyme that really links fat and carbohydrate metabolism in the liver and is very central to the synthesis of fat and cholesterol from sugar, such as fructose that we get in our diet.
So, a part of the paper is on the identification and development of an inhibitors of this enzyme, and then you use it in models of hepatocellular carcinoma. What were the effects of these drugs on hepatocellular carcinoma?
Yeah, so in the paper, we showed that if we genetically remove this enzyme, ATP citrate lyase, that the mice uh, were completely protected and had resolution of the hepatocellular carcinoma. Given this data, we wanted to develop a liver-directed drug that could also manipulate the same pathway. And to do this, we developed the drug by looking at fat and cholesterol synthesis in primary hepatocytes, and found that from a screening exercise, a molecule that really shut down the synthesis of fat and cholesterol in these hepatocytes. And from there, we discovered that this drug was liver selective through an activation mechanism. So it doesn't inhibit fat and cholesterol synthesis in all other cells of the body. It really only targets the tumor and the liver, which is really important from a safety perspective. Um, and what we showed in multiple models was that this drug could slow the tumor growth, and actually not just slow it, it was actually killing the tumors. And, for us, this was really, really surprising because previous studies have shown that blocking de novo lipogenesis will stop the synthesis of fats and cholesterol. And this is needed for when as cancer cells are dividing. They need these fats and cholesterol to make membranes and to make the tumors. But what we noticed with the drug and in the knockout mice is that actually these tumors were being killed. which suggested that the immune system was being engaged to some degree. And this was really, really an exciting finding from the study in that we weren't just putting these liver cancer into quiescence. Actually, we were switching on the immune system to kill the tumors. And we went on to show that this drug really enhances the effects of immune therapy as well as tyrosine kinase inhibitors by promoting this metabolic switch, which really enhances the immune infiltration and causes this tumor killing.
Yeah, that was something I found very, very intriguing in this paper, is that actually manipulating the metabolism, you have an effect on the immune system. And I think that's also bring the possibility with the new drugs that you have, a combination and so on. So so what do you see? How do you foresee now the field to evolve? Because obviously this is very relevant preclinical work. And so now are you moving on this to get closer to the clinic?
Yeah, we're very excited because this is really, I think, the first indication that if you affect tumor metabolism, you're having this huge impact on the immune system and really reactivating the immune system to find these tumors and kill them. So we're very excited by this prospect. The drug is ready to go into phase one clinical trials. We're just looking for the appropriate partner. But there's many, many exciting questions that this also answers. What are the metabolites that the tumor is releasing, potentially to trigger the immune activation? We really focused on this chemokine called CXCL13, which seemed to be attracting these B cells and these tertiary lymphoid structures into the tumor microenvironment. And these structures really act as, a bit of a surveillance mechanism to kill tumors really early on. And they've been linked to reduced incidence of HCC in many areas. So we're really interested to figure out what is the what is the tumor actually releasing that really is key to driving these immune surveillance, and we think that's quite important. And we're also very interested in testing the effects of our drug in combination with existing standards of care such as tyrosine kinase and immunotherapies, because we think that ultimately this could be a big benefit. And we see a great future for the compound actually also in as an adjuvant therapy after resection of tumors. As you know, most people will relapse after cancer resection. And we think that the opportunity to enhance the immune activation is a really great way to prevent so many people from relapsing into liver cancer after resection. And we are really looking, hopefully, trying to move that into the clinic.
Well, effectively. So we'll probably have many more papers to read the next years on this topic. But thank you very much, really, to spending the time with us explaining this beautiful paper in Nature. And congratulations again.
Well, thank you so much for having me, and I look forward to chatting with you again sometime soon.
Be part of the future and join our fight against liver cancer.